Hello everyone. Today let us see about DNA fingerprinting. It comes under the chapter Molecular Basis of Inheritance, class 12. Class 12, Molecular Basis of Inheritance. Under that we are going to see about DNA fingerprinting. So what is DNA fingerprinting? It's a laboratory technique. Okay, first of all, it's a laboratory technique. It is used to determine the probable identity of a person. So, with the help of DNA fingerprinting, you can identify a person. Probable identification of a person based on the nucleotide sequences of certain regions in human DNA. Okay, it is done based on the nucleotide sequences, that ATGC combination. Okay, based on that, that is the base pairing of nucleotides. So, it is based on the nucleotide sequences of certain regions in the human DNA that are very unique to individuals. Okay, even though 90% of the DNA remains the same in all human beings, at least few percentage of DNA differs. That makes individual unique. So that differences are very unique. By that we can identify who is that person. So that is what DNA fingerprinting is, a laboratory technique. It is done in lab conditions. It is used to determine the probable identification of a person. You can identify a person how based on the nucleotide sequences, that is ATGC combination at certain regions of human DNA which are unique to individuals. Okay. Okay, before we go into the technique, the steps, let us see some terms and meanings. Okay, knowing terms and meanings will be helpful in learning the technique. So let us see the terms and meanings. Probe. So when you read the steps, you will be coming across such words. So you should know what is a probe. So a probe is a single stranded DNA or RNA used to search for its complementary sequence in a sample genome. Okay, a small fragment of DNA. Okay, with the help of that, they keep checking the complementary strand in the sample genome. Okay, so probe, it is a single stranded sequence of DNA or RNA used to search for its complementary sequence, to search for complementary sequence in a sample genome. So that is a probe. Okay, another term, tandem. We'll come across another term called tandem. It means repeated nucleotides. Okay, sequence of nucleotides that are repeated. That is that ATGC combination, base pairing, which is repeated many number of times. Okay, and they are in an adjacent manner. In an adjacent manner. Okay, so this is tandem. Tandem means repeated sequence of nucleotides. Okay, some more definitions. Let us see. First is satellite DNA. So it is highly repetitive DNA consisting of short sequences repeated large number of times. Okay, highly repetitive. Okay, it is repeated again and again. The nucleotides, they are repeated again and again. Okay, consisting of short sequences and a large number of times. Okay, that is called satellite DNA. Next word is polymorphism. What do you mean by polymorphism? Poly, many different variants. See, that it is the presence of two or more variant forms of a specific DNA sequence. Two or more variant forms of a specific DNA sequence that can occur among different individuals of a population. In simple terms, it is also known as inheritable mutations in a population. Inheritable mutations. Mutations, sudden heritable changes. Okay, so it is transfer from parents to children and likewise. Inheritable mutations. In short term, polymorphism is said to be inheritable mutations. Okay, presence of two or more variant forms of a specific DNA sequence. Specific DNA sequence, two or more variant forms that is occurring among populations, individuals and populations. Right. So let us now see the steps involved in DNA fingerprinting, the various steps. Actually, this technique was developed by Alec Jeffrey in 1984. Alec Jeffrey, he only started this technique, and the steps involved are as follows. First, isolation of DNA. DNA has to be isolated. Then, digestion of DNA. It is done by the enzyme restriction endonuclease. Then separation of DNA fragments. DNA fragments are separated by the method electrophoresis. Okay, we'll be reading in the further chapters. Okay. Then transferring. Transferring method is also known as blotting. It is separation of DNA fragments to synthetic membranes such as nitrocellulose or nylon. Transferring, this technique is also known as blotting. Here, the separated DNA fragments are transferred to synthetic membranes such as nitrocellulose or nylon. Hybridization. Okay, now that separated and transferred DNA fragments, they are hybridized. It is done by radioactive labeled means radioactive label VNTR, variable number of tandem repeats. So here we use the term tandem. That's what in my initial uh, introduction I told the meaning for tandem. Okay, it is repetition of nucleotides. So variable number of tandem repeats, VNTR. Probe also it told. It's a small segment or fragment of DNA to search for its complementary sequence. So these are all the meanings. So to understand better, first you have to know the terms and meanings thoroughly. So hybridization is done by label VNTR probe. Okay, variable number of tandem repeats. And also be asked for a 1 mark. Variable number of tandem repeats probe, VNTR probe. Finally, detection of hybridized DNA fragments. It is done by autoradiography. Okay, in further chapters and all, I'll be reading about this electrophoresis technique and autoradiography. Okay, so these are the steps, six steps involved in DNA fingerprinting. Isolation of DNA. After that, digestion of DNA by restriction endonuclease. Then separation of DNA fragments. It is done by electrophoresis, a method called electrophoresis under lab conditions. It's a laboratory technique. Initially I told it's a laboratory technique. Then transferring of the DNA fragments, separated DNA fragments were to synthetic means artificial membranes such as nitrocellulose or nylon. It's also known as blotting technique. Then hybridization, fifth one hybridization, it is done by labeled VNTR probe. Finally, detection of hybridized DNA fragments by autoradiography. Right, these six steps you have to remember. So this specific DNA fingerprinting technique can also be asked in a 3 mark question. Oh. Okay, finally we have come to the applications of DNA fingerprinting, the uses of DNA fingerprinting. Okay, so this DNA fingerprinting technique is used as an identification tool in forensic science. Okay, many untoward incident happens like women being exploited. Okay, like something like rape, all these things, so that the criminals can be traced out. Okay, by their bloodshed, hair follicles, skin. Okay, so all these tissues contain DNA. Okay, blood, hair follicles, skin, etc., nail. Okay, So it serves as an identification tool in forensic science, especially to identify the criminals. Then it forms a basis of paternity testing in case of disputes. There is any parental dispute, okay, someone claiming that he or she is my child or something like that. Those disputes can be solved by DNA fingerprinting, DNA matching. Okay, so to solve the disputes, it is useful. Third thing, it is having immense application in the field of genetic biodiversity to determine the populations. Okay, genetic biodiversity and evolutionary biology. Because as we study that inheritable mutation, things like that, okay, it will also lead to speciation, variation. Okay, these things will result in evolutionary biology. So three important uses: identification tool in case of forensic science, forms a basis of paternity testing in case of disputes, has immense application in the field of genetic biodiversity and evolutionary biology. So with that, we come to the end of this video session. Hope you enjoyed learning this. In my next video, let us see about some other topic. Thank you for listening. Kindly subscribe, like, share. Thank you.